Primary production is the synthesis of organic compounds from atmospheric or aqueous carbon dioxide. It principally occurs through the process of photosynthesis, which uses light as its source of energy, but it also occurs through chemosynthesis, which uses the oxidation or reduction of chemical compounds as its source of energy. Almost all life on Earth is directly or indirectly reliant on primary production. The organisms responsible for primary production are known as primary producers or autotrophs, and form the base of the food chain. In terrestrial ecoregions, these are mainly plants, while in aquatic ecoregions algae are primarily responsible. Primary production is distinguished as either net or gross, the former accounting for losses to processes such as cellular respiration, the latter not. Overview. Primary production is the production of chemical energy in organic compounds by living organisms. The main source of this energy is sunlight, but a minute fraction of primary production is driven by lithotrophic organisms using the chemical energy of inorganic molecules. Regardless of its source, this energy is used to synthesize complex organic molecules from simpler inorganic compounds such as carbon dioxide and water. The following two equations are simplified representations of photosynthesis and chemosynthesis. CO2 plus H2O plus light CH2O plus O2, CO2 plus O2 plus 4 hours do S CH2O plus 4S plus 3 hours do O. In both cases, the end point is a polymer of reduced carbohydrate, N, typically molecules such as glucose or other sugars. These relatively simple molecules may be then used to further synthesize more complicated molecules, including proteins, complex carbohydrates, lipids, and nucleic acids, or be respired to perform work. Consumption of primary producers by heterotrophic organisms, such as animals, then transfers these organic molecules up the food web, fueling all of the Earth's living systems. Gross primary production and net primary production. Gross primary production is the amount of chemical energy as biomass that primary producers create in a given length of time. Some fraction of this fixed energy is used by primary producers for cellular respiration and maintenance of existing tissues. The remaining fixed energy is referred to as net primary production. NPP equals GPP, respiration, by plants. Net primary production is the rate at which all the plants in an ecosystem produce net useful chemical energy. It is equal to the difference between the rate at which the plants in an ecosystem produce useful chemical energy and the rate at which they use some of that energy during respiration. Some net primary production goes toward growth and reproduction of primary producers, while some is consumed by herbivores. Both gross and net primary production are in units of mass per unit area per unit time interval. In terrestrial ecosystems, mass of carbon per unit area per year is most often used as the unit of measurement. Terrestrial production. On the land, almost all primary production is now performed by vascular plants, with a small fraction coming from algae and non-vascular plants such as mosses and liverworts. Before the evolution of vascular plants, non-vascular plants likely played a more significant role. Primary production on land is a function of many factors, but principally local hydrology and temperature, the source of energy for photosynthesis. While plants cover much of the Earth's surface, they are strongly curtailed wherever temperatures are too extreme or where necessary plant resources are limiting, such as deserts or polar regions. Water is consumed in plants by the processes of photosynthesis and transpiration. The latter process is driven by the evaporation of water from the leaves of plants. Transpiration allows plants to transport water and mineral nutrients from the soil to growth regions, and also cools the plant. Diffusion of water vapor out of a leaf, the force that drives transpiration, is regulated by structures known as stomata. These structures also regulate the diffusion of carbon dioxide from the atmosphere into the leaf, such that decreasing water loss also decreases carbon dioxide gain. Certain plants use alternative forms of photosynthesis, called crassulacean acid metabolism and C4. These employ physiological and anatomical adaptations to increase water use efficiency and allow increased primary production to take place under conditions that would normally limit carbon fixation by C3 plants. Oceanic production. In a reversal of the pattern on land, in the oceans, almost all photosynthesis is performed by algae, with a small fraction contributed by vascular plants and other groups. Algae encompass a diverse range of organisms, ranging from single floating cells to attached seaweeds. They include autotrophs from a variety of groups. Eubacteria are important photosynthesizers in both oceanic and terrestrial ecosystems, and while some archaea are phototrophic, none are known to utilize oxygen-evolving photosyntheses. A number of eukaryotes are significant contributors to primary production in the ocean, including green algae, brown algae and red algae, and a diverse group of unicellular groups. Vascular plants are also represented in the ocean by groups such as the sea grasses. Unlike terrestrial ecosystems, the majority of primary production in the ocean is performed by free-living microscopic organisms called phytoplankton. Larger autotrophs, such as the sea grasses and macroalgae, are generally confined to the littoral zone and adjacent shallow waters, where they can attach to the underlying substrate but still be within the Photic Zone. There are exceptions, such as sargassum, but the vast majority of free-floating production takes place within microscopic organisms. The factors limiting primary production in the ocean are also very different from those on land. The availability of water, obviously, is not an issue. Similarly, temperature, while affecting metabolic rates, ranges less widely in the ocean than on land because the heat capacity of seawater buffers temperature changes, and the formation of sea ice insulates it at lower temperatures. However, the availability of light, the source of energy for photosynthesis, and mineral nutrients, the building blocks for new growth, play crucial roles in regulating primary production in the ocean. Available Earth system models suggest that ongoing ocean biogeochemical changes could trigger reductions in ocean NPP between 3% and 10% of current values depending on the emissions scenario. Light equals. The sunlit zone of the ocean is called the Photic Zone. This is a relatively thin layer near the ocean's surface where there is sufficient light for photosynthesis to occur. For practical purposes, the thickness of the Photic Zone is typically defined by the depth at which light reaches 1% of its surface value. Light is attenuated down the water column by its absorption or scattering by the water itself, and by dissolved or particulate material within it. Net photosynthesis in the water column is determined by the interaction between the Photic Zone and the mixed layer. Turbulent mixing by wind energy at the ocean's surface homogenizes the water column vertically until the turbulence dissipates. The deeper the mixed layer, the lower the average amount of light intercepted by phytoplankton within it. The mixed layer can vary from being shallower than the Photic Zone, to being much deeper than the Photic Zone. When it is much deeper than the Photic Zone, this results in phytoplankton spending too much time in the dark for net growth to occur. The maximum depth of the mixed layer in which net growth can occur is called the critical depth. As long as there are adequate nutrients available, net primary production occurs whenever the mixed layer is shallower than the critical depth. Both the magnitude of wind mixing and the availability of light at the ocean's surface are affected across a range of space and time scales. The most characteristic of these is the seasonal cycle, although wind magnitudes additionally have strong spatial components. Consequently, primary production in temperate regions such as the North Atlantic is highly seasonal, varying with both incident light at the water's surface and the degree of mixing. In tropical regions, such as the gyres in the middle of the major basins, light may only vary slightly across the year, and mixing may only occur episodically, such as during large storms or hurricanes. Nutrients equals. Mixing also plays an important role in the limitation of primary production by nutrients. Inorganic nutrients, such as nitrate, phosphate and silicic acid are necessary for phytoplankton to synthesize their cells and cellular machinery. Because of gravitational sinking of particulate material, nutrients are constantly lost from the Photic Zone, and are only replenished by mixing or upwelling of deeper water. This is exacerbated where summertime solar heating and reduced winds increases vertical stratification and leads to a strong thermocline, since this makes it more difficult for wind mixing to entrain deeper water. Consequently, between mixing events, Primary production constantly acts to consume nutrients in the mixed layer, and in many regions this leads to nutrient exhaustion and decreased mixed layer production in the summer. However, as long as the photic zone is deep enough, primary production may continue below the mixed layer where light-limited growth rates mean that nutrients are often more abundant. Iron Another factor relatively recently discovered to play a significant role in oceanic primary production is the micronutrient iron. This is used as a cofactor in enzymes involved in processes such as nitrate reduction and nitrogen fixation. A major source of iron to the oceans is dust from the Earth's deserts, picked up and delivered by the wind as aeolian dust. In regions of the ocean that are distant from deserts or that are not reached by dust-carrying winds, the lack of iron can severely limit the amount of primary production that can occur. These areas are sometimes known as HNLC regions, because the scarcity of iron both limits phytoplankton growth and leaves a surplus of other nutrients. Some scientists have suggested introducing iron to these areas as a means of increasing primary productivity and sequestering carbon dioxide from the atmosphere. Measurement. The methods for measurement of primary production vary depending on whether gross versus net production is the desired measure, and whether terrestrial or aquatic systems are the focus. Gross production is almost always harder to measure than net, because of respiration, which is a continuous and ongoing process that consumes some of the products of primary production before they can be accurately measured. Also, terrestrial ecosystems are generally more difficult because a substantial proportion of total productivity is shunted to below-ground organs and tissues, where it is logistically difficult to measure. Shallow water aquatic systems can also face this problem. Scale also greatly affects measurement techniques. The rate of carbon assimilation in plant tissues, organs, whole plants, or plankton samples can be quantified by biochemically based techniques, but these techniques are decidedly inappropriate for large-scale terrestrial field situations. Net primary production is almost always the desired variable, and estimation techniques involve various methods of estimating dry weight biomass changes over time. Biomass estimates are often converted to an energy measure, such as kilocalories, by an empirically determined conversion factor. Terrestrial In terrestrial ecosystems, researchers generally measure net primary production. Although its definition is straightforward, field measurements used to estimate productivity vary according to investigator and biome. Field estimates rarely account for below-ground productivity, herbivory, turnover, litterfall, volatile organic compounds, root exudates, and allocation to symbiotic microorganisms. Biomass-based NPP estimates result in underestimation of NPP due to incomplete accounting of these components. However, many field measurements correlate well to NPP. There are a number of comprehensive reviews of the field methods used to estimate NPP. Estimates of ecosystem respiration, the total carbon dioxide produced by the ecosystem, can also be made with gas flux measurements. The major unaccounted pool is below-ground productivity, especially production and turnover of roots. Below-ground components of NPP are difficult to measure. BNPP is often estimated based on a ratio of ANPP, BNPP rather than direct measurements. Gross primary production can be estimated from measurements of net ecosystem exchange of carbon dioxide made by the ED covariance technique. During night, this technique measures all components of ecosystem respiration. This respiration is scaled to daytime values and further subtracted from NA. Grasslands. Most frequently, peak standing biomass is assumed to measure NPP. In systems with persistent standing litter, live biomass is commonly reported. Measures of peak biomass are more reliable if the system is predominantly annuals. However, perennial measurements could be reliable if there were a synchronous phenology driven by a strong seasonal climate. These methods may underestimate ANPP in grasslands by as much as 2 to 4-fold. Repeated measures of standing live and dead biomass provide more accurate estimates of all grasslands, particularly those with large turnover, rapid decomposition, and interspecific variation in timing of peak biomass. Wetland productivity is similarly measured. In Europe, annual mowing makes the annual biomass increment of wetlands evident. Forests. Methods used to measure forest productivity are more diverse than those of grasslands. Biomass increment based on stand-specific allometry plus litterfall is considered a suitable although incomplete accounting of above-ground net primary production. Field measurements used as a proxy for ANPP include annual litterfall, diameter or basal area increment, and volume increment. Aquatic equals, in aquatic systems, primary production is typically measured using one of 6 main techniques: variations in oxygen concentration within a sealed bottle, incorporation of inorganic carbon-14 into organic matter, stable isotopes of oxygen, fluorescence kinetics, stable isotopes of carbon, oxygen-argon ratios. The technique developed by Gardner and Gran uses variations in the concentration of oxygen under different experimental conditions to infer gross primary production. Typically, 3 identical transparent vessels are filled with sample water and stoppered. The first is analyzed immediately and used to determine the initial oxygen concentration. Usually this is done by performing a Winkler titration. The other 2 vessels are incubated, one each in under light and darkened. After a fixed period of time, the experiment ends, and the oxygen concentration in both vessels is measured. As photosynthesis has not taken place in the dark vessel, it provides a measure of ecosystem respiration. The light vessel permits both photosynthesis and respiration, so provides a measure of net photosynthesis. Gross primary production is then obtained by adding oxygen consumption in the dark vessel to net oxygen production in the light vessel. The technique of using 14C incorporation to infer primary production is most commonly used today because it is sensitive, and can be used in all ocean environments. As 14C is radioactive, it is relatively straightforward to measure its incorporation in organic material using devices such as scintillation counters. Depending upon the incubation time chosen, net or gross primary production can be estimated. Gross primary production is best estimated using relatively short incubation times, since the loss of incorporated 14C will be more limited. Net primary production is the fraction of gross production remaining after these loss processes have consumed some of the fixed carbon. Loss processes can range between 10 to 60% of incorporated 14C according to the incubation period, ambient environmental conditions and the experimental species used. Aside from those caused by the physiology of the experimental subject itself, potential losses due to the activity of consumers also need to be considered. This is particularly true in experiments making use of natural assemblages of microscopic autotrophs, where it is not possible to isolate them from their consumers. The methods based on stable isotopes and O2/A ratios have the advantage of providing estimates of respiration rates in the light without the need of incubations in the dark. Among them, the method of the triple oxygen isotopes and O2/A have the additional advantage of not needing incubations in closed containers, and O2/A can even be measured continuously at sea using equilibrator inlet mass spectrometry or a membrane inlet mass spectrometry. However, if results relevant to the carbon cycle are desired, it is probably better to rely on methods based on carbon isotopes. It is important to notice that the method based on carbon-stable isotopes is not simply an adaptation of the classic 14C method, but an entirely different approach that does not suffer from the problem of lack of account of carbon recycling during photosynthesis. Global as primary production in the biosphere is an important part of the carbon cycle, estimating it at the global scale is important in Earth systems science. However, quantifying primary production at this scale is difficult because of the range of habitats on Earth, and because of the impact of weather events on its variability. Using satellite-derived estimates of the normalized difference vegetation index for terrestrial habitats and sea surface chlorophyll for the oceans, it is estimated that the total primary production for the Earth was 104.9 GTC/1. Of this, 56.4 GTC/1 was the product of terrestrial organisms, while the remaining 48.5 GTC/1 was accounted for by oceanic production. Scaling ecosystem-level GPP estimations based on ED covariance measurements of net ecosystem exchange to regional and global values using spatial details of different predictor variables, such as climate variables and remotely sense FAPA or LAI led to a terrestrial gross primary production of 123 plus or minus 8 GT carbon per year during 1998 to 2005, in aerial terms. It was estimated that land production was approximately 426 GC Ma2 Ara1, while that for the oceans was 140 GC Ma2 Ara 1. Another significant difference between the land and the oceans lies in their standing stocks. While accounting for almost half of total production, oceanic autotrophs only account for about 0.2% of the total biomass. Estimates. Primary production and plant biomass for the Earth. From R. H. Whitaker, quoted in Peter Stiling, Ecology, Theories and Applications. Human Impact and Appropriation, Extensive Human Land Use Results and Various Levels of Impact on Actual NPP. In some regions, such as the Nile Valley, irrigation has resulted in a considerable increase in primary production. However, these regions are exceptions to the rule, and in general there is an NPP reduction due to land changes of 9.6% across global land mass. In addition to this, end consumption by people raises the total human appropriation of net primary production to 23.8% of potential vegetation. It is estimated that, in 2000, 34% of the Earth's ice-free land area was devoted to human agriculture. This disproportionate amount reduces the energy available to other species, having a marked impact on biodiversity, flows of carbon, water and energy, and ecosystem services, and scientists have questioned how large this fraction can be before these services begin to break down. Reductions in NPP are also expected in the ocean as a result of ongoing climate change, potentially impacting marine ecosystems and goods and services that the oceans provide. See also, biological pump, F ratio, productivity, hydrogen sulfide. References